let's start chapter 15. And chapter 15 talks about the special sense. So when we say special sense and when we say general sense, what do we actually mean by that? What are the differences? We talked about it before, right? Yeah, these are the examples, but my question is how the general senses are different from the special sense.
How the information is processed, like the association areas.
Anybody else here? You guys are getting closer. Anybody else want to maybe talk about a little bit more? Yeah? No? Nobody? Really? We talked about it in the lab, right? General sense. When we say general sense, it's all over your body. Like, for example, your touch, right? It's on your skin, touch receptor. But when we say special sense, you have the special organ for sensing that special sense. Like, for example, for vision, you have eyes, where you have the retina, the receptor, which is housed in your eyes, right? For hearing, you have the hair cell receptor. These are housed in your ear, right? For smelling, you have receptor in your nose, not all over your body, right? And then you have what is it called, the taste, you have tongue for that, and that is especially in the tongue. You cannot taste with your skin. Do you taste with your skin? Do you see with your eyes closed? Do you hear with your without your ear? No. But your touch can be any part of the body, can feel the sensation of the touch, right? Okay, that is the differences between the what, that is the most important difference between your what, special sense and the general sense. That's the first thing. Now I want you to understand that for each sense, to be able to sense anything, you have to have the receptor for it, right? When we say we have to have the receptor for it, what does that mean? It means for touch, you have the touch receptor, right? Remember the tactile corpuscle? Yeah, we talked about it when we were talking about the skin. For vision, you have the retina, which is your receptor, and then you have hair cells in your ear for your hearing receptor. So if you do not have the receptor for sensing anything, you would probably not be able to sense that. Not only that, your receptor should be stimulated by that specific information that you are trying to collect. Like, for example, if you think about the sound wave, there are sound wave which could be from 0 hertz to like 100,000 hertz, but can you hear all of them? No, you only hear the sound which your body is allowing you to hear. So basically your hair cell can only be stimulated by 20 hertz to 20,000 hertz, and that is the only sound wave that you can actually what, that's the only thing you can actually hear. Similar to that, there are so much of like electromagnetic wave, there are different type of the wave around, but do you see all of them? You do not. You only see very small spectrum of your visual wavelength of light, right? So what does that mean? It, whatever your receptor is allowing you to receive, that is only the information you can actually receive. Does it make sense? Now you know the receptor, and if you do not have the receptor, you cannot actually take that signal. Like, for example, you have eyes but your retina is degenerated, would you be able to see? No, you are going to be blind actually. Yeah, so the receptor is very important. Without the receptor, you cannot actually take any information from the environment. Okay, so now with the receptor you can actually receive the signal. That receive the signal is called the what? the sensation. You take the signal, you sense it. Like for example, because you have retina in your eye, you can probably see this slide right here, right? Now, after that, if you think about the sensory receptor, this sensory receptor right here, what is it telling you also? That you can see it, but what you can see, what is the interpretation? When we say the sensation and the interpretation difference, what do we mean by that? Sensation is you are receiving a lot of what? Light hitting in your retina, right? That is the sensation. Now what is written here, what does that mean? That is what the interpretation that is happening inside your brain, that is called the what? Perception, right? The perception. So there is a difference between the sensation and the perception. And we already know that the receptors we have in our body are different types based on the location, based on the stimulus type and all this, right? You guys already know about this one, right? Type of the receptor, which based on the like sensation or stimulation. Like for example, if you think about the mechanoreceptor, that is your what? Respond to touch, pressure, vibration, stretch, right? These are all by your mechanoreceptor. And then you have your thermoreceptor. We did talk about it a little bit elaborately last time, right? So when we say right now, if you like 70-degree, if it increases to 80-degree, it is received by the thermoreceptor as warm, right? Feeling of hot. However, if 70-degree turn into 200-degree, do you think are you going to receive that signal with the thermoreceptor? Which receptor is going to be stimulated then? Nociceptors. Very good. Because nociceptors is sensitive to pain causing stimuli like extreme heat or cold, excessive pressure, inflammatory chemicals. But what about the photoreceptors? Photoreceptors is present inside your eye, the retina, right? And what about the chemoreceptors? Your taste, your smell, these are all happening because of the chemicals that are coming from your environment and getting what? Stimulating your receptor in your nose, in your tongue, and all this. Everyone knows about this one too, right? Classification based on the location. If the receptor is outside of your body, then you call it exteroceptor. Like for example, receptor for touch, receptor for vision, receptor for hearing. All of these are receptor for temperature, pressure, pain. All of these are what? Exteroceptors. But when we say interoceptor, what do we mean by that? Or the visoreceptor, what do we mean by that? Visoreceptor, what do we mean by the visoreceptor? Internal, like for example, if there is anything happening to your viscera or organ, that is the information you are receiving by the visoreceptor. For example, your stomach ache, your headache, right? All these are coming, all these are what? You can sense that because of the interoceptor. What about the proprioceptor? Like all of your muscles stretch, ligament stretch, bone, tendon, right? These are coming with the proprioceptor. And as I told you already, when we say the special sense, you have the special organ to sense that specific stimuli. So what are the special sense? Vision. For vision, what do you have? Your eyes. And for taste, you have tongue. For smell, you have nose. For hearing and equilibrium, you have the ears. So now, do you guys know that 70% of your receptors, so all the receptors that we have been talking about, like your smell, your taste, your touch, 70% of all receptors are actually in your eyes. So basically, even if you are not thinking about it, your eyes are taking lots of information from the environment. You have 70% of the body's receptors in your eye, and that the cerebral cortex that you have, half of it actually processing the information you are taking with that receptor in the eye. So maybe you are probably not aware of it just by looking, just not even you were not even aware of it. You are like, just you have your eyes open, you are taking a lot of information, and 50% of your cerebral cortex is actually dealing with the all the information that you are receiving, with 70% of the what receptor in your eyes all the time. That is why this vision is actually a little bit different. Do you know, we have been talking about, every time we talk about the stimulation, what do we actually mean by the stimulation? Stimulation happens due to the, what happens chemically? Depolarization, right? Stimulation happens due to the what? Depolarization. But your eyes, as long as you have your eyes open, what does that mean? Your cells are what? Always depolarized, right? So always depolarized. Is it true? Little bit, I want you to think about it. So you're 70% of the stimulus in your eyes, right? Actually, let's not talk about it right now. When we talk about how the vision is happening, then I'll talk about it. It's a little bit different, because all the stimulus is stimulating the receptor by generating the graded potential and finally depolarizing it. Only for the eye, only for the vision, it is starting with the hyperpolarization. Only for the eyes, it is starting with the hyperpolarization. Okay, so why, we will talk about it, but just I want you to know how this vision is actually very different from other senses. Okay. Okay, so now we already know about the different parts of the eyes, but what are the accessory structure of the eye? Everyone knows about the eyebrow, eyelid. Eyelid is also called what? Palpebrae, right? And then when we say conjunctiva, what do we mean by the conjunctiva? So we know that we have that transparent layer, we call it cornea. So outer layer of the cornea, you also have a mucus layer that is called conjunctiva. And this conjunctiva are actually different types, like palpebral conjunctiva, underside of the eyelid, and bulbar conjunctiva, white of the eyes, and there is also conjunctival sac, which is the space between this. And everyone knows about the lacrimal apparatus, right? Lacrimal gland, which produces your what? Tears and all this. And there are also 6 extrinsic eye muscles. What are the name of those eye muscles? So how many rectus do you have? You have 4 rectus. What are those? Superior, inferior, medial, and lateral, and 2 oblique. What are those? Superior oblique and the inferior oblique. Very good. So this is something you guys already know. Where is your levator palpebrae? There is another muscle which is called the levator palpebrae. What is the levator palpebrae? Helps with the close and opening of your eyelid, because eyelid is what? Also called palpebrae. And then you have the levator palpebrae. This is your eyebrow. I don't want to talk about this. You guys understand, it's pretty straightforward. But do you know where is your lacrimal gland is located? We talked about it very well in the lab. Where? On the lateral side or the medial side? On the lateral side. Superior or inferior? Superior lateral. Very good. So now in the lacrimal gland, this is the one which produces and secretes the what? Tears. So tears enters the conjunctival sac via the excretory duct of the lacrimal gland, and then tears flow down and across the eyeball. Tears enters the lacrimal canaliculi at opening called lacrimal puncta, and tears drain into the lacrimal sac, and from the lacrimal sac tears empty via the nasolacrimal duct into the inferior meatus of the nasal cavity. So think about that. Okay, so everyone knows about these muscles right here, right? What are those muscles? Which one is what? Which one is this one right here? lateral, medial rectus, superior rectus, inferior rectus, superior oblique, and inferior oblique. Now you have to know this chart very well. So let me tell you, let me actually make sure you understand this. So your lateral rectus and medial rectus, these are kind of like name is telling you what do they do, right? Lateral rectus moves the eye laterally and medial moves the eye what? Medially. But what do you think about your superior rectus? Superior rectus and inferior oblique does the same thing. What do? They elevates the eye. So for looking up, you have to use the what? Superior rectus and the inferior oblique. The who is helping you move down? Your inferior rectus and superior oblique. So in your exam, I did ask that question, and students always make mistake because there are rectus is superior rectus is doing the opposite thing, right? Superior rectus, what is the superior rectus helping you with? Depression, looking down, right? And then inferior rectus helping you with the what? Elevating, looking up. Okay, so make sure you do not make that mistake. And also know that only optic nerve is associated with taking the information of the visual information. However, for eye movement, there are other cranial nerve like abducens, oculomotor, and trochlear. These three are associated with the eye movement. So in your exam, if I ask you which of the following is not associated with the eye movement, and if I give you abducens, oculomotor, trochlear, optic, what are you gonna choose? Optic, because optic does not help you with the eye movement. Instead, it helps you with the what? Taking the visual information from your retina to the brain. Okay, so now this is something everyone knows, right? Conjunctivitis, pink eye, very highly contagious. As I have been telling you, the mucus layer, outer mucus layer of your eye is called the conjunctiva, and it can be inflamed, and then what can happen? Your eyes can be red and irritation may occur. So pink eye, very highly contagious because it is an infection by the bacteria or viruses. I asked question about that as well. Okay, pink eye, so know about that. Okay, so we know that when we were talking about the eye, and also whenever we have done the cow eye dissection, we have seen that there are two cavities. One cavity is what? Posterior to your lens. One cavity is what? Anterior to your lens. Anterior to your lens, what do we call it? Anterior cavity. And the posterior to your lens, what do we call it? Posterior cavity. So in the anterior cavity, what type of fluid do they have? Aqueous humor. And the posterior cavity, what type of fluid do they have? Vitreous humor. Very good. So now let's talk about this aqueous humor. What is that aqueous humor? First of all, I want you to understand any fluid in your body is actually the what? Filtration product of your what? Blood, right? So now if you think about your aqueous humor forms by filtration from the capillaries in the ciliary processes. So how they are making the aqueous humor? By filtration from the blood. And this aqueous humor flows from the posterior chamber through the pupil into the anterior chamber, and some also through the vitreous humor. And this aqueous humor is reabsorbed into the what? Venous blood by the scleral venous sinus. Okay, so now this aqueous humor which helps you what? Keeping your eyes wet and moist, right? If you do not have that, what is going to happen? You will have dry eyes. So now this is the structure of the eyes. I'm sure everyone knows already. Everyone knows where is your cornea, where is your ciliary body, iris, pupil, and then your lens, right? And then you have three layers. You have the outermost layer, which is called the what? Choroid. And then you have the, what is the next layer? The first outermost layer is called the sclera. Everyone forgot. I'm saying the wrong thing and you guys are not telling me anything. The outermost, the white layer is called the what? Sclera. We talked about it many times in the lab, right? And then underneath that, you have that what? Very vascular, right? That is called the choroid. And underneath that, you have the what? Retina. Very good. So now in your exam, if I ask you, so you have this three layer. The outermost layer is the what? Fibrous layer. So if I ask you the sclera, is it fibrous, vascular or retina? You're gonna say it's fibrous. What about the cornea? Cornea is also the part of the what? Fibrous layer. What about your iris, ciliary body and the choroid? These are your vascular layer. What does that mean? It means they have a lot of blood circulation in there. And then what about your retina? That is your pigmented layer. And then there is also a neural layer. When we say neural layer, what does that mean? They are made up of the neurons. So do you guys remember the differences between the iris and the pupil? When we say iris, what do we mean by the iris? The color part of your eyes is the what? Iris. And that iris is attached to the what? Ciliary body. When ciliary body pull your iris, you will have your what? Pupil dilated. And then when ciliary body what? Relaxes, then your pupil is going to be constricted. Now the question become, when does that happen? Whenever you have your sympathetic activated, you want to be alert, you want to see far. That is why you want to have your pupil dilated or constricted. During sympathetic, dilated. And during the parasympathetic, you do not want to see more. You do not want a lot of what? Lot of light enters into your eyes. What do you want to do then? You want to make sure that your pupil is what? Constricted. So I did ask question about that in your exam too. Like, make sure you know what happens during the sympathetic and what happens during the parasympathetic. So do you guys all remember about this part right here? We know that we have these three layers. The outermost layer is the sclera, choroid, and the retina. So the retina, the whole retina has the what? Receptor for vision, like the red rod and cone cell, right? However, there are some specific part that you have to know very well. Number one is called the optic disc. This is the place where you are receiving all the neurons, all the axons are going through the optic nerve toward your what? Brain. So this optic disc is the very important part right here. The reason this is the important part right here, they are the one who is receiving the information from all over your retina except this optic disc. If any light falls on the optic disc, do you think you will be able to see that? You cannot see that. That is why the optic disc is also called the blind spot. So now whenever you look at the retina, it absorbs the light and prevents its scattering and phagocytizes photoreceptor cell fragments and stores vitamin A. So why, when we say for vision, the whenever we were like very young, like very small, they're like, take a lot of vitamin A, eat vitamins A because it's good for your eyes. Everyone knows that, right? Why it is important for you? How does that help? We're going to talk about it. But just make sure that your retina also stores the what? Vitamin A. So as you can see right here, the structure of your retina, you have these cells that you see, the outermost layer, this outermost layer of the cells, these are called the what? Photoreceptors. The purples are rods, most of them are actually rod, and these yellow are the what? Cone. And then these are the bipolar cells and these are the what? Ganglionic cells. So as I told you, your photoreceptor, the outermost cell, rod and cone, bipolar cells right here, and then the ganglionic cells, these are the innermost layer. Their axons gather at the optic disc and form the optic nerve. Now, we did talk about the optic disc, that optic disc does not receive any information from that specific part because that is the blind spot. However, do you know which part of your retina is helping you to see the best? Which part? We talked about it in the lab. There is a part in your retina which takes the information, visual stimulus very effectively. What is the name of that part? Macula lutea. It contains the fovea centralis. This is the central pit, and this has the highest proportion of the cone, area of the sharpest vision. Okay. So now what if this macula lutea is degenerated? What is going to happen? You will start losing the vision. Okay, this is the very important part of your retina. And then there is the peripheral retina. All over your retina, mostly they all only have the what? Primarily have the rods, functionally most effectively in the low light. Who remembers about the differences between the rod and the cone? What is the difference between the rod and the cone? Anybody can tell me anything about the rod and the cone? Discussed in the lab. What is happening? What is the difference between the rod and the cone?
Cones detect light, and rods detect or the cones detect
Color,
yeah,
and
rods
rod
detect light.
detects light. Yeah, very good. So now as you can see the retina right here, this is the optic disc, the blind spot, and this is the macula lutea, the very important part of your what? Very important part of your eye. So now detach retina. If for any accident, if for any reason the retina is detached, what is going to happen? Your retinal detachment condition where pigmented and neural layers separate, allowing jelly-like vitreous humor to seep between them and deprives retina of the nutrient. What is going to happen? You will have floaters, curtain, flashes of the light, decreased vision, can lead to permanent blindness. Usually happens when retina is torn during the traumatic blow to head or sudden stopping of the head during the movement, treated with the prompt surgery. Okay. Have you ever heard of the glaucoma? What is the glaucoma? So what is glaucoma? What happens during the glaucoma? So we know that you have continuous circulation of the what fluid? What is the name of that fluid? Aqueous humor. What if that aqueous humor is not circulating properly? Instead they started what? Building up and creating pressure. That is when you will have the what? Glaucoma. Condition in which drainage of the aqueous humor is blocked, causing the fluid to back up and increase pressure within the eye. Pressure may increase to dangerous level and compress retina and optic nerve, leading to blindness. What is the symptoms? The early symptoms are there are some few early signs, but late signs include seeing halos around the light and blurred vision. Detection: intraocular pressure determined by the directing puff of the air at cornea and measuring amount of the corneal deformation. Test should be done yearly after age 40. Yeah. No. Lacrimal is producing it. We want to give it back to your venous sinus, scleral venous sinus. You want to give it back to your vein so that it goes back to the blood, right? You create it from the blood and then you give it back to the blood, like all the fluid in your body. Okay. So now what is the treatment? Eye drops that increases the rate of aqueous humor drainage or decrease its production, and laser therapy or surgery. Macular degeneration, very what? This is the reason for blindness, right? Physical deterioration of the macula lutea, leading cause of the blindness in developed countries, may be associated with the diabetes, ocular infection, hypertension, eye trauma. Risk factors are genetics, race, smoking, and loss of the visual acuity in the center of the visual field, and diminishes color perception, and floaters. No cure, but can have surgery or take blood vessels growth inhibitor. Now, do you know that you have a lens in your eye, and that lens is very flexible? What do we mean by the flexible? Flexible. Even though it is biconvex, not biconcave, it is biconvex. When we say biconvex And when we say biconcave, what do we mean by that? This is the biconcave, right? But yours is what? Biconvex. However, this biconvex lens, you can change the shape and the size to be able to what? See at different distance clearly, right? Because you have the what? Ciliary body, which is attached to the lens. So biconvex, transparent, flexible, and avascular. No vasculature, so no blood circulation to that place. Changes shape via the suspensory ligaments and ciliary muscles to precisely focus light on the retina. And lens anatomy, anterior lens epithelium and the lens fiber, and lens fibers are continuously added, so lens becomes more dense, convex, and less elastic with the age. It does become less elastic with the age. So what does that mean? What does it tell you? What does it tell you really? So as we age, our capacity to see at different distance clearly, what changes also? It becomes deteriorates, it becomes bad. Yeah. Blind spot is you are collecting all the neurons from there. That is the blind spot. You are collecting the neurons, but you are not receiving at that specific place any more visual stimulation. Okay. And everyone is kind of familiar with this term cataract. When we say cataract, what do we mean by that? It's like another layer in front of your eyes, right? The clouding of the lens, consequence of the aging, diabetes mellitus, heavy smoking, frequent exposure to the intense sunlight. Some are congenital, and also what happens? We know that what is the reason for our very crystal clear cornea? Because we have the protein called crystalline, but the crystalline protein clump. So that is why it turns into what? Cloudy instead of clear. Vitamin C increases the cataract formation. Lens can be replaced surgically with the artificial lens. So now I'm sure you guys are very familiar with this type of the eye problem, right? For example, I have myopia, severe myopia. I cannot see without the corrective lens very far. Okay, so when we say what does that actually mean? It means, like, for example, if you think about your eyeball, if you think this is your macula lutea, what is supposed to happen if the what your wave, the visible light waves are coming and falling on top of the what? Macula lutea, you can see it clearly. But what if you have a very big eyeball? Instead of falling it over here, it's falling right here. So do you think you'd be able to see it clearly? You are supposed to see it clearly right here. So now you cannot see it clearly. So what do you have to do? You have to wear a lens which help you to what? Move it over, yeah, so that you can see it clearly. And that is called the what? Myopia. And what is happening in the hyperopia? Hyperopia is what is happening in the hyperopia. This is normal, this is where you are supposed to have it fallen, macula lutea. But when we say hyperopia, what is happening in the hyperopia? Instead of falling it over here, it's falling, you have the smaller eyeball is falling over here. Then do you think you'd be able to see it clearly? You cannot. So in that case, you have to use the biconcave biconcave lens to see it clearly. Okay. So now let's talk about, I made a mistake. So myopia is corrected with the biconcave lens, and then hyperopia with the convex lens. And what about the presbyopia? Presbyopia, have you ever heard of that? So we know that we have our lens, which is very, very what? Flexible, right? It can see, it has the capability to see at different distance clearly, right? When we lose that accommodation, that is when you have the presbyopia. It usually as we age, the what? that is what kind of we get. And have you ever heard of the astigmatism? What happens? It's the unequal curvature in your different parts of the cornea or the lens. So correct it with the cylindrically ground lenses or the laser procedures. So a lot of the time, a lot of us can have few conditions together at the same time. A lot of the time you might have to wear a glass which not only is trying to fix your myopia but also probably is trying to fix your hyperopia, right? Or also maybe trying to fix your what, astigmatism. Okay, so how much do you remember? Let's see, what type of receptor do you find in your eye? Photoreceptor. Which layer of the eye contains the cornea? Your cornea, outermost layer. What is the name of that layer? Fibrous layer. Vascular layer is the next one, right? What creates the blind spot? What creates the blind spot? Optic disc. Why? Because with the optic disc you are sending all the neurons, axons, right, toward your brain, right? Okay, so now let's talk about the photoreceptor a little bit in detail. So when you look at the photoreceptors, we know that these purples are actually your rods and this yellow is your what? Cone. So you have the synaptic terminal, cell body, inner segment of each joint cells and the outer segment light receiving region, and contain the visual pigments, photopigments that change shape as they absorb the light and constantly replaced by the to maintain the vision. So as I have been talking about the rod and cone differences, we already know that you have a lot of rod, less cone. And what is the function of the rod? Rod, non-color vision, and cone is the what? Color vision. And rod has the very high sensitivity and cone has low sensitivity. So you can only see the color in the bright light, not in the dark. And rod has the low acuity, but cones has the high acuity. And rods are very like numerous and cones are less numerous. So you find the rod mostly in the peripheral retina and cones mostly in the central retina. So have you ever heard of the term color blindness? That is happening due to the what? The problems in your cones, right? Usually a genetic disease, and also it's X-linked, that means it's sex-linked. So usually males are more prone to have that color blindness. As many as 8 to 10% of the males have some form of the color blindness. The most common type is the red and green, in which either red cones or green cones are absent. Depending on which cone is missing, red can appear green or vice versa. Rely on different shades to get cues of the color. So now let's talk about something important. So what is the key light absorbing molecule in your eye? The main light absorbing molecule in your eye is called the retinal. Retinal. What do we call it? Retinal. That retinal combines with the opsin 4 protein. This is the what? Your visual protein. Okay. Opsins, and form the visual pigments. And these opsins convert photon of the light into the electrical signal. We know that how we receive any information, we receive the information always with the electrical signal, which is hyperpolarization or the depolarization. So this is actually synthesized from the vitamin A. That is why your parents tell you to what? Take vitamin A so that you have good vision. So now you know why do you need vitamin A. How does that help you see? So there could be 4 different type of the opsins. One is called the rhodopsin. Rhodopsin is only for the rod, and there are 3 more for the cones. And these 3 more are called the green, blue, and the red. And cone wavelengths do overlap. So some wavelength may trigger more than one cone, and enabling us to see variety of the hues of the color. So now we only have the how many cones? 3 cone, red, green, and the what? Red, green, and the blue. But we see burgundy, we see purple, we see orange, we see sky blue, we see different colors. How do we see that? The reason we see that, it's what, it's based on the combination. So yellow light stimulates the red and green cones, but if more red are triggered, then we see orange. So, and another thing to tell you, not everyone see the same color in the same way. Just to let you know, we see color in different way too, based on the what, our genetics, our cones, the presence of the cones, number of the cones, all these things. Okay, now this is the important part. So you have this retinal, usually it's the band form, which is the 11 cis. This is the cis. When we say cis, it has all of this in a same direction, right? Cis. So this is the band form. The first thing is happening to be able to, when they get stimulated, they are gonna be straight. When they get straight, we call them trans. Okay, so this is your cis pigment, cis retinal pigment, and this is your trans retinal pigment. So conversion of the band to the straight initiates a reaction that leads to the electrical impulses along the optic nerve. Okay, so now this is what I wanted to tell you. So whenever you have your photo, like whenever there is dark, no light is coming, that is when you depolarize. So what does it mean? You are actually receiving only the signal of the what? Not seeing. Most of the time you actually see whenever the light comes in. So that is why your photo, like inhibition is happening by the what? Depolarization. Okay, and excitation is happening by the hyperpolarization. Is it making sense? Most of the cases, all of the other cases, always excitation is happening with the what? Depolarization. Only for the eyes, you have your excitation happening starting with the what? Hyperpolarization. Because whenever the light is coming, what does that mean? It's just normal for them. Okay, you are only sensing it whenever there is no light coming. Okay, that is the information you are trying to take. Yeah. This is only for eyes. Yeah, only for eyes. It's a little bit different, because when we say it is starting with the hyperpolarization. So you see when it's hyperpolarized. Okay, it's not excited, that's when you see. But here, what is happening, like you start with the depolarization starts whenever there is no light coming. Does it make sense? It's opposite to all other receptors. This is very different. That is why I was telling you, your eyes are different, your eyes are special. Yeah, even though you are starting with the depolarization, the inhibition, but the finally at the end, what do you want to do? You want to make sure that is hyperpolarization. But here, whenever you want to see something, what do you want to do? You start with the hyperpolarization, but at the end, what do you want to do? You want to generate the action potential, right? But you start with the hyperpolarization. That is the difference. Okay, so now this is important. So now let's talk about the dark adaptation and the light adaptation. So what happens when we say dark adaptation? What do we mean by that? So return to the sensitivity to low light levels after the bright light. So bleached rods must regenerate rhodopsin and may take 20 to 30 minutes to see well. So right after you went to sleep. So before you go to sleep, what do? You turn off the light. So after you turn off the light, for first few minutes, probably you do not see anything. But if you keep your eyes open, after sometimes you start seeing things even at the dark. That is called the what? Dark adaptation. And similar to that, you also have to what? Become adapted to light. So every time you become adapted to light, what does that mean? Your sensitivity of the retina should actually increase or decrease? Decrease, because in the light you want to decrease the sensitivity of the retina. But what is happening? So process of the adjustment from the low light to the bright condition, pupils constrict, but cones initially were overstimulated and takes about 5 to 10 minutes for all full adjustment. And usually decrease the sensitivity of the retina. Yeah.
I know, like older folks, they usually struggle to see in the dark. Is that because like the
Rhodopsin, yeah.
Is that like degrade over time, or like
That's a great question. Let me answer you like next on Wednesday, because I have some answer, but I'm not 100% sure about it. Okay, let me just make sure I understand first, and then I'll let you know. Okay, so now what is the visual pathway to your brain? How do you see? You do not see in your eyes. You just take the information with your eyes. Then when you see, when you can actually send it to your occipital lobe, that's when you see, right? So how is it going? It's going from the photoreceptor to the bipolar cell to the ganglionic cell, then optic nerve, then optic chiasm, then optic tracts, then lateral geniculate, nucleus of the thalamus, then visual cortex of the occipital lobe. That's when you see. Yeah. No. Yeah. Okay. So one thing I want you to know right here is like this visual, we know the pathway that we have been talking about. One thing we did not talk about right here, whenever they are going through it, they also have to go through optic chiasm. When we say optic chiasm, they have the superior colliculi and the inferior colliculi. Which one they are actually going through is the superior colliculi. Okay. Okay, so which ion channels change their status when light strike a photoreceptor? Did I tell you the name of that? I did not. That is a cyclic GMP. Let's talk about that. I forgot to talk about it. So the cyclic GMP gated ion channel open. Okay, cyclic GMP gated. GMP means what? Guanosine monophosphate. That is cyclic guanosine monophosphate gated ion channel. Okay, so which type of the photoreceptor allows the color vision? Cones, yeah, good. Now, does anybody have any question about the eyes? No question about the eyes? Okay, so let's talk about the ear, another one. So everyone knows about the ear, right? Your auricle or pinna, helix, lobule, earlobe, and then what is the name of this canal right here? External acoustic meatus. And then you start your middle ear with the what? Your tympanic membrane, and then you have this middle ear. In the middle ear, you have the three smallest bones in your body. We call them malleus, incus, stapes. At the end, you have the what? Oval window, and then from the oval window you are starting your what? Inner ear. In the inner ear you have the what? Semicircular canal and the what? Cochlea. This cochlea actually houses the receptor for hearing, which is called hair cell. Hair cell or hair cell? Hair cell, not hair cell. Okay, hair cell, it's like hair cell. Okay, so now from when the hair cell is stimulated, that is the information you send to your brain by which nerve? What is the name of the nerve? Vestibulocochlear nerve. Okay, good. So now if yeah, this is something we were just talking about already. So we know that there is one tube right here, that is the, what is the name of this tube right here? External acoustic meatus, and in the middle ear you have another tube which is called the pharyngotympanic tube, also called the eustachian tube, connects middle ear to the nasopharynx, usually flattened tube but can be opened by yawning or swallowing to equalize the pressure in middle ear cavity with external ear pressure. We talked about it in the lab, right? Remember, whenever the flight takes off, what happens? You start losing the equilibrium between the pressure of the external acoustic meatus and the pharyngotympanic tube. So what you can do to make sure you can equalize the pressure by yawning or yeah.
I'm pretty sure the same thing happens
Mm-hmm.
like
Yeah, you start losing the equilibrium of the pressure. Yeah. So tympanic membrane cannot vibrate efficiently whenever there is no equal pressure on both sides and not equal, and sounds are distorted. So so you have these three smallest bone in the middle ear, right? What is the name? What are the name of those? What are the names of those? Malleus, incus, stapes. These are also called the three auditory ossicles. Three auditory ossicles. So now your tympanic membrane, whenever you look at your tympanic membrane, have you ever heard of that? That the eardrum is infected or eardrum is, so many things can happen to that. So this is a normal healthy eardrum, tympanic membrane. And look at this right one. When this is infected, right? Ear infection. Okay. Okay, so now in the internal ear, you have the labyrinth, bony labyrinth and the membranous labyrinth. So the bony labyrinth is the system of the tortuous channels and the cavities that worm through the bone. And what about the membranous labyrinth? Series of the membranous sacs and the ducts contained in the bony labyrinth filled with the potassium rich endolymph. So everyone knows about the cochlea, right? Cochlea is a very important part of your ear because that is what which houses the what? Your hair cells, not hair cells, hair cells. And also they have the three cavity. What are the name of those cavity? Vestibulo, huh? What is the name of those? What is the name of the first one, second one, third one? Who remembers those? Scala vestibuli, organ of Corti, cochlear duct. In the cochlear duct you have the organ of Corti, and then you have the scala tympani. We'll talk about that. But now first of all I want you to understand the spiral organ or the organ of Corti has the what? Receptor for the hearing, and then that is what is actually the part of your cochlear duct, right? And then you have the vestibule contain the macula, receptor for the equilibrium. Receptor for the equilibrium, where do you have that? In your semicircular canals, right? The saccule is continuous with the cochlear duct and the utricle is continuous with the semicircular canal. So in the semicircular canal you have the ampulla, that is what is basically the receptor for the equilibrium, and large area of the duct of each canal that houses the equilibrium receptor region called the crista ampullaris. Okay, respond to the angular movement of the head, angular movement of the head. So as you can see the cochlea right here, and then you have all this cochlear nerve, spiral ganglion. Oh, that's not important, let's not talk about it. But this is the cochlea right here. This is something very important, right? Right here, this is the what? Scala vestibuli, your cochlear duct. In the cochlear duct, what do you have? You have the basilar membrane, organ of Corti, hair cell, tectorial membrane. Remember all those different parts. And then this is the what? Scala tympani. Scala tympani is also called the what? Everyone forgot. Remember whenever I was talking about how hearing works? Scala tympani is the what? Trash can. Why this is called the trash can? Because your hair cell can only be stimulated by 20 hertz to 20,000 hertz. Whatever is not stimulating your hair cell is going to be disposed in the scala tympani. Okay. Okay. In the organ of Corti, you have the basilar membrane, organ of Corti, hair cell, tectorial membrane. Now, how the hearing is happening? We know that first of all, with the acoustic, external acoustic meters, the sound wave should come and what? Vibrate your eardrum, and then your sound wave should be amplified through these ossicles, malleus, incus, stapes, and then go through the oval window, and then where does it go to? Scala vestibuli, then go to organ of Corti. After going to organ of Corti, what do? They stimulate your hair cell. After stimulating the hair cell, they will have the friction with the tectoral membrane. That is the information you send with the vestibular cochlear nerve to your brain so that you can hear the sound, right? Now whatever cannot stimulate will go and get disposed in the vestibulo-scala tympani. Yeah, that is how the hearing occurs. So conduction deafness, blocked sound conduction to fluid of the internal ear. Causes including impacted earwax, perforated eardrum, otitis media, which is the ear infection, right? Otosclerosis of the ossicles. And there could be sensory neural deafness. So one is the conduction deafness. If in the conduction pathway, if there is any problem, that is your conduction deafness. And what about the sensory neural deafness? If any sensation, like in the neurons, in the damage in the hair cells, damage in the vestibulocochlear nerve, damage in the organ of Corti. So these are the sensory neural deafness, damage to the neural structure at any point from the cochlear hair cell to the auditory cortical cells, typically from gradual hair cell loss. And potential treatment, cochlear implants that convert sound energy into electrical signals are effective for congenital or age or noise cochlear damage. So as you can see, we have the three different parts of the three different type of the receptor. You have the semicircular canal, you have the crista ampullaris for the equilibrium, which is the rotational acceleration, and the vestibule, utricle and saccule, equilibrium, head position relative to gravity. And then the receptor region is the macula, and your cochlea, which is the hearing, the spiral organ. So now, as I have been talking about, the vestibule, which is the macula, helps with the what? Equilibrium, head position and the linear acceleration. And the semicircular ducts, which is the crista ampullaris, is taking the equilibrium, rotational acceleration. And now the question becomes, we know that your hair cell can be stimulated by 20 to 20,000 hertz. However, you can hear the best from 1500 to what? 4000 hertz. Okay. So now what do we mean by the pitch and the quality? So when we say the pitch, that's the perception of the different frequency. This is the frequency. So different frequency will help you to understand different pitch. But how do you understand the loudness? How do you think about something? How do you perceive something loud? Because the amplitude, okay, the frequency is helping you to determine the pitch, and the amplitude of the frequency is helping you to what? Understand that as a loudness. Okay. So height of the crest, amplitude perceived as the loudness, and subjective interpretation of the sound intensity measured in decibels. Normal range is 0 to 120 decibels. Normal conversation is around 50 decibel, and threshold of the pain is 120 decibel. So when it's like really high, it's gonna start feeling like pain. Severe hearing loss can occur with prolonged exposure above 90 decibel. So hearing in balance is the function of your ear, right? Language skills tied to the ability to hear well, and then few ear problems until 60s, when deterioration of the spiral organ becomes noticeable. And hair cell number decline with age. I asked you a question about it. What is the presbycusis? So presbycusis is the what? Loss of high pitch perception occurs first, type of the sensory neural deafness. Have you ever heard of the tinnitus? Sometimes you hear the sound without any what? Stimulation. That is the what? Tinnitus. Sometimes I hear some kind of sound without any noise. Yeah. Yeah. Yeah, it is neurological. It is neurological. It is, yeah. This ringing, buzzing, and cockling sound in the ears in the absence of the auditory stimuli, because the cochlear nerve degeneration and inflammation of the middle or internal ear, side effects of aspirin. And there is another one, don't mix this up. Meniere syndromes also has the same thing. You hear the sound without the stimulation. However, it comes also with other thing like vertigo, nausea, and vomiting. Okay. So that is why this is the difference with the tinnitus with the what? Meniere syndrome. So that's why I asked you a question about it during the exam. Don't write like tinnitus is the Meniere syndrome whenever it's coming with both vertigo, nausea, and the vomiting. Okay. Treatment is anti-motion sickness drug in mild cases, or surgical removal of the labyrinth in the severe cases. So what did you learn so far about the hearing? Where are the ossicles and what role do they play in hearing? In the ossicles are in the ossicles. You have the three ossicles. In the
Yeah,
middle
yeah,
ear.
yeah.
What is the function of the ossicles? Amplification. Amplification of the sound wave. What are the three parts of the spiral organ of the inner ear? Spiral organ. What do they have? Basilar membrane, hair cell, tectorial membrane. What kind of head movement is detected by the semicircular duct? Rotation, right? Okay, now you also have the two chemical what senses, like your smell and taste. Do you know your smell and taste? And your taste is also coming from your smell. Your taste is almost like 80% of the smell. Both of them are what? Chemical receptor, right? Chemical receptor. Both of them are chemoreceptor, and they are both really what? Dependent on each other. Not your smell is dependent on taste, but your taste is really dependent on the smell. Without the smell, taste is like bland, right? Okay, so smell and taste, olfaction and gustation. So how can you smell? Where is the smell organ? Organ is the what? Nose, and the membrane inside the nose that has the receptor for the what? Smell. So complementary senses that let us know whether a substance should be savored or avoided. Chemoreceptors are used by these systems. Chemicals must be dissolved in the aqueous solution to be picked up by the chemoreceptor. Smell receptors are excited by the chemicals dissolved in the nasal fluid. Taste receptors respond to the chemicals dissolved in the saliva. So smells may contain hundreds of the different odorant. That is something important. Do you know some people are very sensitive to odor and smell, and some people are not? Because you can actually have 10,000 to 1 trillion odor receptor. However, not all of them have the same. It depends on your genetics and all these things. I'm not very sensitive to smell, but there are some people who can actually slight changes in the environment, they could like there is something going on, somebody is eating some kind of special vanilla cookie or something, they could tell, but I cannot tell. I think I'm very like, yeah. No, it's just how many like human, so humans have 350 different odor receptors, but not everybody has all of them. Yeah. Yes. So each respond to one or more odorant, protein respond to one or more odors. Each odor binds to several different receptor types, and each receptor cell has only one type of the receptor protein. And pain and temperature receptors are also in the nasal cavities, respond to irritants such as ammonia or can smell hot or cold, like chili peppers, menthol. And tasties, as I told you, 80% smell. So if your nose is blocked, you are not gonna actually taste the food very well, because if nose is yeah, it tastes bland. And mouth also contains the thermoreceptor, mechanoreceptor, nociceptors. These days kids love eating all those hot Cheetos and all those Takis. Do you know why do they like it? Even though it's so hot, why do they like it? The pain sensation is actually not by your taste, it's by your pain sensation. Sometimes whenever you have some kind of what is it called, some kind of soreness somewhere, and then sometimes you just try to pinch it or like do it to feel that pain, but you feel good about it, right? I don't know if you ever have that happen. You're scratching the painful area just to induce a little bit more pain. I know have you ever had that? I did that. I love it sometimes. I actually love it sometimes. So this is very interesting. This is very interesting. So spicy hot food can excite pain receptor in mouth, which some people experience as pleasure. So that's what I'm telling you. It's even though this is actually they sense it as a pain, but it's just enjoyable.
It's the exact opposite. It actually feels like my like pain, like pinching or like.
Uh-huh.
And so my mom jokes that I must be allergic to spice because like my nose starts running, my eyes get all watery, and I
Oh,
get
okay.
all hot, like red.
But you don't like that, right? You don't like that.
But my
Okay.
brother's the exact opposite. He'll buy thousands of hot.
Oh my goodness, my daughter is too. I'm like, oh, why are you eating all these colors and chemicals? Like, really?
I actually did a lot of research on this because I got really addicted to like the flavor of chili peppers
Mm-hmm.
and stuff, and so there's like a chemical in there. So not only does it give you this kind of adrenaline
Mm-hmm.
rush, but there's also a chemical in there that
Mhm.
stimulates dopamine and serotonin.
Yeah,
And your
yeah, it does.
metabolism and
Yeah,
all of that.
yeah, yeah.
It's just a
So
fun
let me tell you my story. I used to like hot food a lot. Like hot means chili pepper hot. I used to like it so much because the place I grew up in, we had a lot of food which is like it comes with a lot of different spices. It's like very flavorful. But after coming to USA, especially when I started staying alone, eating a lot outside, becoming like started eating a lot of bland food, now I cannot take this spice anymore. And I'm like, oh, I'm sorry, I'm sorry. I'm so sorry. Yeah, so when I go to my house, like they're like it's so tasty, so yummy. I'm like, oh my goodness, so hot, I cannot take it. Yeah, so yeah. But I have friends like I had some American friends like same food. I'm enjoying it so much, and they're like, it's like not at all spicy for me, and they're like already started crying. Yeah, so you imagine like what I'm trying to tell you right here. It's all based on your receptors, adaptations, and all this. Yeah, that's all about this chapter. So when you come on Wednesday,